Thanks for coming. I'm Chris Gobler. I'm the Director of Academic Programs here for the School of Marine and Atmospheric Sciences at Stony Brook University. And you're attending a Friday night seminar as part of our spring lecture series. So this is the second in an installment of 4. We'll have another seminar here in this room the first Friday in May and the first Friday in June. With the next one being by Bill Wise about the history of the menhaden fishery in New York waters. But tonight, thank you for coming to the Stony Brook Southampton Coastal and Estuarine Research Program's 8th Annual Environmental Symposium. It's a long title, but I thank you for coming. I hope you had some time to see the student posters outside, and I'll talk a little bit more about those posters as I go through my presentation. And joining me tonight and presenting will be Dr. Brad Peterson. So I'll be kicking things off and then he'll be cleaning things up, so to speak. So I think I'm just going to go ahead and get started. As you see, SCERP has been around for about 8 years, and the overall objective of SCERP is to protect and restore our coastal ecosystems. So that is to do things like minimize the impact of anthropogenic stressors on these coastal ecosystems and maximize the distribution of fisheries and foundational species such as fish, filter-feeding bivalves, and eelgrass. And specifically, as the name indicates, we're involved in research. So we're conducting research that's identifying environmental threats and what their root causes are, and then also, at the same route, coming up hypothetically or potentially with solutions for some of these environmental problems. So that's the first major objective. And then the second one is being fulfilled tonight. You all are here, and part of what we're trying to do is communicate our results to the public, to government agencies and non-government agencies, so that the research doesn't just sit on our desks or go into a journal, but perhaps can be used to make positive changes in our coastal ecosystems. Tonight I'm going to talk to you about 2 topics, and then Brad Peterson will be talking to you about the final topic. We're talking about the impacts of land use on our estuaries, a process called coastal ocean acidification, and then Brad Peterson will talk to us about prospects for restoring our coastal ecosystems. And the first topic I'm going to cover tonight is nitrogen, and specifically nitrogen in the South Shore bays of Long Island. Now, the New York State Department of Environmental Conservation is the watchdog for our coastal waters, and specifically has a list that it generates every year and compiles through the years. It's called its Impaired Water Body List. For those of you who are in the field, the 303D list. And this list, bodies of water are put on this list when they violate New York State standards for problems with things like oxygen or pathogens. And historically, particularly on eastern Long Island, if there was a water body put on this list, generally put on for things like a fish kill and low oxygen associated with that, or pathogens, and generally it was just very small tributaries. And then further west, places you can imagine like the Gowanus Canal, for example, that has serious environmental problems. But recently that's been changing. We're at the point now, for those who aren't aware, that the entire South Shore Estuary Reserve of the state of New York, which extends from Hempstead Bay in the west and all the way to the shores of eastern Shinnecock Bay, about 1,000 feet from where we're standing, is now listed on New York State's Impaired Water Bodies list. This is 100 kilometers of contiguous waterway. And this is not what's been going on historically. This is a very recent occurrence and a very recent change. This figure shows the kilometers of South Shore shoreline that's been listed through the years. And as you can see, with the listing of the Western Bays, and then in 2010, the listing of Great South Bay, Moriches Bay, and Shinnecock Bay, we're now where we are today with 100 kilometers of shoreline defined as impaired. And the listings for most of these areas, the reason they're impaired, the reason the state lists is the overloading of nitrogen and the associated algal blooms. And one of these areas you can learn more about tonight, specifically Hempstead Bay. There's a poster being presented by graduate student Ryan Wallace. He's doing an intense study of this area, and if you want to learn more about that, I'd encourage you to speak to him after the presentations are done. But right now, as it stands, that South Shore estuary represents 1/4 of all of our shorelines on Long Island. And I told you they're listed there because of reasons of nitrogen. So we know what the problem is, and actually I would argue that we have the solutions in front of us as well, and that's what I'm going to talk to you about right now. So when you look at nutrients in any given water body, what's really important, and it's important that I make this point very clearly, one size does not fit all, and the nutrient that is most important for a given water body varies depending on where you are. So I'm going to talk to you right now about the South Shore Bays of Long Island, but everything I'm going to tell you is not the case for other waterways. So for example, the Peconic estuary, the situation is different. So we're talking right now about the South Shore Bays. There are plenty of parallels with other waterways, but I want to be clear that I'm talking about these South Shore estuaries. So very recently, the Nature Conservancy commissioned scientists to construct a nitrogen budget for Great South Bay, the largest bay within the South Shore Estuary Reserve, and to determine what is the biggest source of nitrogen. We know that's the problem. Where's it coming from? Wastewater, fertilizer, the atmosphere. And I can point to you within a 50-mile radius where we're sitting right now, there are different estuaries in New York where each of those is the most important nutrient. So this was for Great South Bay. And the answer, by and large, for Great South Bay was wastewater. 70% of the nitrogen coming from land going into this estuary is coming from wastewater. And now what's very interesting about that, for those of you who know the watershed of Great South Bay, the whole western extent of Great South Bay is on a sewage treatment plant that discharges the sewage directly into the ocean, essentially saying that half of that 71% is coming from only half of the watershed, which means when you move to places like Moriches Bay or Shinnecock Bay, none of that nitrogen is being pumped into sewage treatment plants. So all of the nitrogen is coming from homes. The septic tanks discharge into the groundwater, and the groundwater makes its way into the South Shore Bays. So while the number is 70% for Great South Bay, that number is substantially larger, likely for Moriches Bay, Shinnecock Bay, and Quantuck Bay. Now, concurrent with this report, Suffolk County has been monitoring groundwater very carefully via the Department of Health Services and looking at levels of nitrogen in our groundwater. And they just finished and released a report that came out. It's in draft form right now, but the information in that report is kind of surprising. Specifically looking at changes in the groundwater in Suffolk County that have occurred in only an 18-year period, going from 1987 to 2005. And right here, what I'm showing you are the two upper levels of the aquifer on Long Island. The glacial aquifer, which is the one that discharges, has the biggest influences our bays, and then the Magathy, which is deeper than that. And the one that we've always assumed would be pristine and we could never impact. But you can see over this very short period of time, only 18 years, that in the upper glacial aquifer we've had a 40% increase in levels of nitrogen and a 200% increase in the Magathy, to where the levels today are greater than they were only in 1987. So these are pretty radical and rapid changes that are happening. And again, in the Magathy, that was an aquifer we thought that we'd always have for ourselves. We never really could affect that one. Thankfully, there's one more that's deeper, but we can only guess that that will be affected similarly going forward. So how did this happen? In only an 18-year period, how did we have a 200% increase in nitrogen in our groundwater? Well, a lot of it links back to our population growth. As I'm sure you're all aware, the 2010 census data just become available. And, we're not growing at the rate we did in the mid-20th century, but the population of Suffolk County continues to grow. And some areas are built out. And some people would tell you, well, we don't need to worry about development anymore because there's no more room to grow. And that is certainly the case, actually, along the Great South Bay watershed. But further out east, actually, the growth rates of our population are outpacing those of the rest of Suffolk County, where we have more than double the growth rate in towns like East Quogue and Hampton Bays, which are towns that are situated right on the South Shore Estuary Reserve. So those populations continue to grow at a substantial rate. And when that happens, that's when our populations grow, that's how nitrogen levels can get changed. And this is an example from Suffolk County that was just released. This is one of several case studies they showed, and I'll just show you this one. This is Bicycle Path. This is in the town of Brookhaven. But there's many other cases we can point to that look just like this. So in 77, this is this parcel of land. The white indicates undeveloped, where it's orange is just medium density housing. So fairly sparse, not a lot of housing. By 2004, this area was built out with medium density housing. Concurrently with this change, overlying that land, the nitrogen levels changed drastically from a level below 1 milligram per liter, which you can consider maybe natural, pristine, but over this same exact time period, the levels of nitrogen increasing more than 7-fold. And this is, again, a pattern for one case study, but this has been shown in many places across Long Island. In fact, this has been known for many years. So what happens when we have the levels of nitrogen increasing by 40 to 200% that are in the groundwater and development increasing the nitrogen loading into our South Shore bays? Well, we know one definite consequence is harmful algal blooms. And that's an issue globally. I was involved in a project that was initiated by the EPA to come up with a consensus on what we know about nutrients and harmful algal blooms. And it's hard to get consensus among scientists, but the consensus is that nutrient loading is promoting harmful algal blooms. And that's certainly also the case here locally. So looking back to 1980, this is before we had that big increase in nitrogen, we had a situation where there were no documented harmful algal blooms on Long Island in 1980. Robust shell fisheries and seagrass meadows. We know that our population's increased, we know the nitrogen loading has increased, and we've had an outbreak of harmful algal blooms in many different regions. So you're looking here at a picture, nice view, we're somewhere over here. Here's the Shinnecock Canal, Shinnecock Inlet, Shinnecock Bay, nice blue water, Peconic Estuary. But that water in recent summers during the months of July, August, September, has been turning this sort of color with blooms of the dinoflagellate Cocclodinium polycricoides. Last summer the blooms broke out in July, much earlier than usual, and lasted into almost October. And creating this sort of streaky-like pattern that discolors the water, but unfortunately that's not the only impact. I'll talk about more about that in a second. But we know that these blooms are promoted by nitrogen. This is some experimental work with a series of experiments where bottles are enriched with nitrogen, and we follow the growth of multiple types of phytoplankton and see which of the population responds most consistently to nitrogen. In this case, it's that red tide species, Cocclodinium, that's responding either to all different sorts of nitrogen compounds or some specific organic nitrogen compounds. And if you want to learn more about this, I'd recommend you see the poster by Dr. Tong and Florian Koch specifically on these blooms, and they can tell you not just that they're promoted by nitrogen, but their ability to actually very rapidly cause mortality in finfish, shellfish, zooplankton, and also phytoplankton. Another red tide species we now have in Long Island is known as Alexandrium. You're looking here not at the South Shore bays, but this is actually Northport and Huntington Bay along the North Shore. This is a species that has been present for a long time, but never has caused a problem until 2006, where we had our first occurrence of PSP, paralytic shellfish poisoning, in New York. And since then, it's occurred in 4 of the past 5 years, where this whole system has been closed down because of blooms by this red tide dinoflagellate that contains the poison saxitoxin and makes shellfish toxic. And research that's been conducted by Teresa Hattenrath, who's also giving a poster here, is shown here, where she did experiments where she'd take water from one of these red tides and amend it with nothing, phosphorus, or 4 different types of nitrogen compounds, and then measure the levels of the toxic algae or the toxin within the algae. And the results are clear. If you increase the amount of nitrogen loading, you get more toxic cells and more toxin. And in the same study, Teresa was able to isolate the cells that form these blooms and use an isotopic technique to determine that the nitrogen being assimilated by these cells is derived from wastewater. So again, the wastewater connection is present there. Now again, I'm talking about the North Shore, but Teresa's also done surveys throughout Long Island. And what's striking is that she's yet to find a location on the South Shore where these cells don't exist. So when she looks in the springtime, the cells are always there. And in some locations, places like the Forge River, which we know is highly impacted on the South Shore by nitrogen, at levels that are enough to cause a problem. And so Northport's not unique, and we know there's many things happening in Northport. There's a sewage treatment plant right there. There's dense housing. But we see the potential for what's happening there to occur in many places along the South Shore of Long Island. And again, Teresa's presenting a poster tonight about this particular species and another dinoflagellate out there known as Dinophysis, which contains another class of toxins known as DSP toxins. And I'd encourage you, if you're interested, to talk to her about that later this evening. Also on the harmful algal bloom front, we're probably most familiar with brown tides. They've been around here for the longest period of time of all these species, since 1985. These are aerial photographs of the 2008 brown tide bloom in Great South Bay. I see Carl LeBou is here from the Nature Conservancy, and he can tell you the ill effect that this bloom had on their clam restoration efforts in that estuary. Recently, we actually sequenced the full genome of this organism. I'm not going to give a talk about genomics tonight, but I will just give you some insight that we learned from looking at the genome of this organism and what it can tell us about what's happening in our bays. Because we looked at this species and its genome, but we also looked at other species it's co-occurring with. What you're looking at here is a time series plot from Quantuck Bay, located between Shinnecock and Moriches Bay, and it shows you the intensity of the blooms in 2007, 8, and 9. And what we did, in addition to tracking the cell densities, we also collected a sample and generated something known as a metaproteome. And by doing that, we're able to figure out, well, what other species of phytoplankton is this brown tide competing with, and therefore, how is it built differently genomically? Because for these 6 other species of phytoplankton we identified, their genomes are also available. So how do their genomes align with the conditions we have today in 2010 in our estuaries? I won't go into all the details, but suffice to say, when we look at the brown tide and compare it to these competing phytoplankton, genetically, genomically, it's built to use organic nitrogen compounds and organic carbon compounds better than these competing phytoplankton. It's also has the genetic machinery and capacity to do very well under low light conditions, much more so than these other species. So what does this have to do with nitrogen loading? Well, we know that the more nitrogen we have coming in to our estuaries, particularly the areas that don't circulate well, that promotes higher levels of organic nitrogen, higher levels of organic carbon, and also lower levels of light and higher levels of turbidity. So you've seen the levels of nitrogen go up, and what I'd say is that by increasing these levels of nitrogen, we're transforming our estuaries into systems that are now ideally suited for the genome of Oreococcus, and we think maybe some of these other harmful algal bloom species as well. And if you want to learn more about the brown tide and its genome and genomic work, you can see the poster of Elise Walker and talk to her about her work on some of the genes in that genome after the presentation. And I'll just briefly mention that we also have nutrient loading in our freshwater systems and have problems with that. Here's a headline from the fish kill that occurred in Mill Pond in 2008, and we know that nutrients are driving these systems as well. And again, we have Matt Harkey, who's presenting a poster on the effects of nutrient loading in the freshwater systems and maybe what can be done to remediate that. So in 1980, we had a situation where we had no harmful algal blooms. We've changed the density of our housing along the South Shore. We've seen increases in groundwater levels of nitrogen between 1980 and what we have today. And you can see what's happened since then. All these different harmful algal blooms have shown up on our coastal waters over the last 2 decades. And I've just shown you there are clear links between all of them and the loading of nitrogen. So what are we going to do about this? Who feels like this? Some days I feel like this. And that is one option, of course. But I think the answers are right in front of us. In fact, the studies that I've mentioned in the beginning give us some of the answers. Here's a really nice model that Suffolk County put together that essentially says, you choose how much nitrogen do you want in your groundwater. And once you've decided that, then you should choose how closely you want your houses put together. These different lines are for when you have quarter-acre zoning, half-acre zoning, 1-acre zoning, 2-acre zoning. And essentially, by choosing how many houses and how many septic tanks you have per square acre, you can decide what you want your final nitrogen levels to be. Importantly, in the same study, if you look at these dashed lines, that's showing what happens if you add fertilizer to the scenario. And if you look at that, what's essentially showing is that while fertilizer certainly increases the nitrogen load, it's a very small effect relative to the number of septic tanks that you have in that area. And that should make sense, at least it now makes sense to me intuitively. A septic tank is a chronic nitrogen source 365 days a year, whereas fertilizer is something that's put down once, twice, maybe 3 times a year. So you can see it's not a big effect here. And in the same study commissioned by the Nature Conservancy, they looked at the effects of what if you had people just stop fertilizing? How does that affect the nitrogen load to Great South Bay? And here's the answer. Whether you have everybody fertilizing as they do today or a 90% reduction, hopefully you can see the decrease. It is there, but it's not substantial. So besides fertilizer and besides housing densities, another thing that can be thought about is septic tanks. As I mentioned, once you get past the middle of Great South Bay, the whole eastern end of the South Shore of Long Island, all the homes have septic tanks, and septic tanks are discharging nitrogen on coastal waterways. Well, again, the Nature Conservancy's recently commissioned study shows that if you you can put those septic tanks on sewage treatment plants. That would be a very big effort, obviously, but you can significantly reduce the nitrogen load by over 60%. Alternatively, there are now technologies to use alternative septic systems, and they looked at that in this study and showed you can also get a fairly substantial reduction. And this was using an average septic system upgrade. There's some that are just as effective as sewage treatment plants. So this nitrogen loading can be put in reverse with the right attention. So just talking about nitrogen on the South Shore, I mean, the science now shows that fertilizer is a small problem. Surface runoff is a small problem, and I wish we could have said that 12 years ago. There's a management plan for the South Shore estuaries on Long Island. That whole management plan is built around surface runoff because we know that it puts pathogens into our estuaries, but it's not affecting the nitrogen loads. The atmosphere, at least in the South Shore bays, importantly that we're location specific with this, is not a big problem. The problem is household waste. And so if you wanted to mitigate that, certainly there's a lot of battle yet to be fought with regards to development on the eastern end of Long Island, and therefore lower density is going to give you lower nitrogen loads. And with regards to places that are already built up, upgrading septic systems can certainly reduce the amount of nitrogen going into these systems. Okay, I'm going to transition away from nitrogen now to the second part of my presentation and talk about something known as coastal ocean acidification. So change of pace, take a deep breath. Good. And think about climate change, which is another issue that we need to be well aware of when it comes to our coastal ecosystems. And I think when a lot of people think about climate change, the biggest thing that people think about is global warming, right? And that's almost synonymous with a lot of people. Higher temperatures due to the greenhouse effect. And that certainly has happened. We've had an 8% increase in global temperatures over the past century. But that's a small increase relative to what we've seen with regards to the increasing levels of carbon dioxide in our atmosphere, a 40% increase, and also within our oceans, a 40% increase in acidity. This is a figure showing both the changes in CO2 that are happening over only the past few years on this planet, and the levels are going up consistently. You can see now exceeding the 390 level. We'll soon be at 400 parts per million. We were at about 280 parts per million at the dawn of the Industrial Revolution. And you can see the rate at which the increase of CO2 occurs continues to increase as well. And when we have higher levels of CO2 in our atmosphere, that's rapidly transferred to our upper ocean, and that's affecting the pH of our oceans. This figure shows the dawn of the Industrial Revolution. through into an era we'll never see, obviously. Well, we'll never see any of this, but the year 3000, and takes a pessimistic view of humanity that will simply combust all of our fossil fuels on the planet, and then shows what will happen with regards to CO2 in our atmosphere, and importantly for us as we think about our estuaries, how our surface ocean pH will change. So the pH of the ocean's already declined by 0.1 units and will decline potentially by up to 0.7 units in future decades. This increase in CO2 reacts within seawater to undergo a series of reactions, the most important one being that they eventually lead to carbonate ion being transferred out of, being converted into bicarbonate, and that when we have more CO2, we're having lower levels of carbonate in our seawater. And that's a problem for organisms like coral or any other organism that wants to make calcium carbonate shell. And when we think about our fisheries here in New York State, there's a lot of organisms that make calcium carbonate shell. In fact, 4 of our top 5 fisheries are calcifiers. And going forward, we know that calcifying for them will be more difficult. And amongst these calcifiers, one group of organisms we're focused on, been looking at very carefully, has been the larvae of bivalve shellfish. Adult shellfish spawn, they're broadcast spawners, and release larvae into the water column that are there for about a 3-week period. And we're learning very quickly that these larvae are very sensitive to changes in water chemistry, and specifically changes in carbon dioxide. And we also know already that small changes in these larval survivals can have big impacts on adult populations. So all of this research has been conducted by Stephanie Talmadge. I won't call her Dr. Talmadge yet, but she'll be defending her PhD on these results in only a couple of weeks. And she's done a fabulous job at pioneering all of this work right here in Southampton. Published this paper in the Proceedings of the National Academy of Sciences just last fall. And she looked at how changing levels of CO2 affect these bivalve larvae, and considered not just what's going to happen in the future, but she also considered what happens if you look at the CO2 levels that we used to have on this planet.